All right, let's dive in. Today, we're looking at how artificial intelligence is completely changing how we understand diseases like cancer. Think about it like this. For a long time, we've been trying to figure out this incredibly complex disease by looking at a single kind of blurry snapshot. But what if we could see it in a stunning 3D movie? See it from every single angle, all at once? Well, that's what we're going to explore, how AI is giving us this incredible new dimension of sight.
So this is the big question, right? This is what's driving this huge revolution in medical research. It's not just about piling up more data. It's about getting the right kinds of data. And this is the crucial part, having a way to see how it all fits together. That's where the real breakthrough is happening.
Okay, so to really appreciate how amazing the solution is, we first have to get a handle on the problem itself. And with something like cancer, what we're really dealing with is a massive, incredibly complicated data puzzle.
Historically, researchers would zero in on just one layer of biology. Maybe they'd only look at the genomics, the DNA blueprint. And look, that's super valuable information. But it's kind of like trying to understand a whole city by only looking at the subway map. You're missing the buildings, the people, the traffic, all the stuff that makes it a living, breathing system.
So naturally, scientists tried to combine all these different layers of information. But when they did, they slammed right into three fundamental roadblocks that have, for a long time, made it almost impossible to see the full picture.
Let's break these down real quick. First, you've got data heterogeneity. This just means you're trying to compare apples and oranges and maybe a car engine. The data from genes looks totally different from data about proteins. Second, missing data. It's really expensive and hard to run every single test on every patient, so your big data puzzle ends up with a ton of empty spots. And finally, technical artifacts. You can just think of this as noise or static on the line. Tiny little differences in how samples are processed in different labs can create false signals that hide the real biology we're looking for.
So how in the world do we solve this puzzle? Well, the very first step is to start gathering all the pieces you need on purpose, right from the get-go, so you can actually see the whole picture.
And this brings us to a really important term, multi-omics. This is the whole strategy of collecting all those different layers of biological data, but from the exact same patient. It's a huge shift in thinking. We're moving away from looking at just one piece of the puzzle to trying to assemble the entire thing all at once. The goal is to create one single high-resolution view of the disease.
And these are the key layers we're talking about. You've got genomics, that's the master blueprint, the DNA. Then you've got transcriptomics, which are the instructions being actively read from that blueprint at any given time. Proteomics are the actual machines, the proteins that are carrying out the instructions. And finally, metabolomics covers the chemical reactions that happen as a result of all that activity. Each layer tells a totally different but absolutely vital part of the story.
Okay, so we've done it. We've collected this absolute mountain of multi-layered data. We have all the pieces for our 3D movie. But this just creates an even bigger problem, doesn't it? How do you possibly process and understand this fire hose of information? You need an engine that's powerful enough to find the patterns hidden inside all that complexity.
And yep, you guessed it, that engine is deep learning. This is the technology that can take all that massive, messy, multi-layered data and boil it down into something that makes sense, something we can actually use.
One of the real stars of the show here is a type of AI called an autoencoder. The easiest way to think about it is as the ultimate data summarizer. It takes in literally tens of thousands of data points from all those omics layers, squishes them down into their most essential features. That's the latent representation. And then it tries to rebuild the original data just from that little summary. If it can do a good job, it means that compressed summary must contain the most important signal-rich information. It found the good stuff.
But wait, it gets even smarter. We can add something called an attention mechanism. This basically stops the AI from looking at everything equally. Instead, it learns to figure out what's important. So, for making a specific prediction, it might learn that, hey, this signal over in the protein data is like 60% important, but this other signal in the DNA is only 10% important. It's like giving the AI a smart spotlight to shine on only what truly matters for the question you're asking. So we've got the comprehensive data, and we have this powerful AI engine to make sense of it. What's the payoff? Well, this is where it all comes together. This is the path to what we call precision medicine.
This workflow you're seeing here shows the entire process in action. It's pretty amazing. Over on the left, you start with a big, diverse group of patients. The AI model then crunches all their multi-omics data to find hidden patterns, and it clusters them into new, much more precise disease subtypes. From there, it can figure out the specific molecular things driving each subgroup, and ultimately,
generate a personalized prognosis for one single patient.
So let's just nail this down. It's a three-step process. First, the AI redefines the disease itself. It moves past old classifications to create these new subgroups based on what's actually happening at a molecular level. Then, it pinpoints the exact biological signals, the biomarkers, that define those groups. And finally, it uses those specific biomarkers to make a prediction for an individual patient. It's a journey from massive data down to a single personal insight.
This quote from the research really just says it all, doesn't it? At the end of the day, the goal isn't just to publish cool papers or build fancy AI. It's about taking all of this mind-boggling molecular complexity and turning it into clear, useful information that a doctor can actually use to make a better decision for their patient. That's the whole point.
But as exciting as all of this is, there's still a gap, a pretty big gap, actually, between the research lab and the doctor's office. So we need to talk about the final hurdles we have to clear to make this a reality for everybody. To make this happen, we basically have to build a bridge from academic research over to clinical practice. You've got the high-powered models on one side, and on the other, you have real patients and doctors who need tools they can actually trust. And spanning that gap means solving a few more key challenges.
First up is the black box problem. Sometimes the most powerful AI models are also the hardest to understand. So we need what's called explainable AI, so a doctor can see why the model made a certain prediction. You have to be able to trust it. Second, validation. A model that works great on data from one hospital might completely fail on data from another. We need really tough, standardized testing on all kinds of different patient groups. And finally, there just isn't that much of this high-quality data out there. So to solve this, clever new methods like federated learning are popping up, which allow models to learn from data across multiple hospitals without any of that sensitive patient data ever leaving its home base.
And all of that brings us to the final, and one of the most important question. We've seen the incredible potential of this technology to turn that blurry snapshot into a full 3D movie. But the final challenge isn't just a technical one. It's about building systems that are not only powerful, but also transparent, reliable, and at the end of the day, worthy of our trust with something as precious as our health. Thanks for watching.